Hello, everyone, and welcome to today's live broadcast. Can AI-powered diagnostics revolutionize antimicrobial stewardship? Machine learning-based prediction of antimicrobial resistance in short and long-read sequencing data. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by OpGen and Aries Genetics, an OpGen group company revolutionizing how we address the global challenge of antimicrobial resistance with innovative genome technologies and AI-powered bioinformatics. For more information about our sponsor, please visit their site at opgen.com. So let's get started. I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click the Send button. If you have any trouble seeing or hearing this presentation, simply report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd now like to introduce our presenter, Dr. Arne Materna, a Managing Director and the CEO of Aries Genetics, an OpGen group company. For a complete biography of our presenters, please visit the Biography tab at the top right side of your screen. Dr. Materna, welcome. You may now begin your presentation, sir.
Hello, thank you for tuning into this presentation. My name is Arne Materna and I'm the CEO of Aries Genetics. And over the course of the next 30 minutes, I will speak about how machine learning, when combined with sequencing-based solutions, can overcome key limitations in infectious disease testing and surveillance. A few words up front about Aries Genetics. We are a Vienna-based startup founded in 2017, and together with Curetis, we are part of the OpGen group of companies. Together we fight against antimicrobial resistance through cutting-edge molecular microbiology. While OpGen and Curetis deliver regulatory-approved molecular diagnostics for antibiotic-resistant pathogens, Aries Genetics is where we harness genomics and innovative machine learning approaches to address the global threat of antimicrobial resistance, or AMR. Let's begin with a few facts on AMR. While the attention of many of us quite rightfully is on the impact of the current COVID-19 pandemic, another severe pandemic is on the rise. Antibiotics, which we all have come to rely upon as effective treatment of otherwise often dangerous and sometimes deadly infections, are slowly but steadily becoming ineffective. We see a global increase in pathogens that prove resistant against standard and even last resort antimicrobial therapy. With already tens of millions affected by AMR and dozens of billions of euros per year in direct healthcare costs, the most severe consequence we face are over 700,000 fatalities each year. That is over 2,000 per day. By some estimates, these numbers are expected to rise to 10 million per year by 2050, meaning the AMR epidemic would eclipse cancer as the leading cause of death. The problem of AMR is exacerbated by the challenge of choosing the right treatment. Predicting how an infected patient will respond to antibiotic therapy is a hard problem, influenced by factors such as the genomics of the pathogen that is causing the infection. As a consequence, only a minority of patients are receiving appropriate treatment, a fact that has become apparent once more in the overtreatment of COVID-19 patients with antibiotics to quell bacterial co-infections. In case you wonder why overtreatment is a problem, it is probably worthwhile to take a look into what is actually causing AMR. As any microbial ecologist or microbiologist will explain you, antimicrobial compounds or antibiotics are produced naturally by microbes themselves in order to compete with other microbes inhabiting the same ecological niche. In response to these compounds, microbes evolve the very mechanisms that cause resistance and give us trouble in the care of infectious disease patients. Inappropriate antibiotic use, for example, overuse or simply using the wrong antibiotic for a pathogen, then selectively enriches for microbes that have evolved such resistance mechanisms. And whenever this situation is met with imperfect hygiene and infection prevention, then we face the risk of enriching for and spreading antibiotic-resistant or even multi-drug-resistant pathogens. And that in turn requires increased antibiotic use, fueling the cycle. The question therefore is, what can be done to break this vicious cycle leading to AMR? On one hand, we'll rely on the pharmaceutical industry to keep developing novel forms of antibiotic therapy. The challenge for pharma thereby is that new drug development becomes a race against the evolution of resistance mechanisms. This, of course, is a very tall order. While an encouraging number of 70 drugs is currently in different stages of clinical and preclinical development, the WHO just noted in its recently released 2020 report on antibiotic drug candidates that this number may not be high enough to tackle AMR, as currently over 80% of newly approved drugs can be readily rendered ineffective by existing resistance mechanisms. This does, however, not mean defeat. The improved antimicrobial stewardship can help us regain the advantage over AMR. Antimicrobial stewardship refers to technologies and interventions that promote the optimal use and avoid the overuse of antibiotics. Now, why is this important? As I tried to allude to when addressing the cause of AMR, appropriate use of antibiotics was shown not only to improve patient outcomes, but also to reduce the emergence of antibiotic resistance. And this is where technology can help us make the difference. In order to inform and promote appropriate antibiotic therapy and to enable antibiotic stewardship, we need, on one hand, solutions that diagnose or predict resistance to enable a targeted therapy with appropriate antibiotics, as well as solutions that enable the global surveillance for AMR to allow us to detect outbreaks, to counter them with infection prevention measures, and to empirically inform first-line antibiotic selection. Let's take a look at the technologies that are in the clinic today. The standard of care in infectious disease diagnosis are bacterial cultures, which are routinely used for the identification of infective agents and the determination of suitable antibiotic treatment options. Whenever a culture is positive, meaning if pathogen growth is present, then antibiotic susceptibility testing, also referred to AST, is performed in order to determine which antibiotics are effective and which will likely be met with resistance. While these culture-based methods are being considered the gold standard, they are often labor-intensive and may miss difficult-to-culture pathogens. Turnaround time ranges from 1 to 3 days or more, which is long considering that patients with acute infections require urgent care often within hours. This lack of sensitivity and speed is addressed by modern molecular tests relying on PCR. Leading PCR panels, such as the tests available on the Curetis Univero system, achieve very high sensitivities and can return results in a matter of hours, even pushing the limit below 1 hour. However, these systems can only screen for up to a few hundred biomarkers at best. This means each assay, which has to be individually developed, validated, and potentially approved, can cover a limited number of pathogens, and for each pathogen, only a limited number of genetic markers for AMR. Given that today the number of genetic markers for AMR is measured in the thousands, the question has to be, is there a system that could simultaneously and comprehensively screen for any pathogen and all known genetic markers for antibiotic resistance? And what if the system would not only report the detected AMR markers, but also emulate the advantages of culture by predicting whether detected pathogens are susceptible or resistant to antibiotic therapy? This is where next-generation sequencing, or NGS, enters the picture. NGS is increasingly adopted by clinical labs, and its utility, for example, for microbial typing in epidemiology is already undisputed. It is the first technology that, thanks to an enormous sampling depth, is not only very sensitive, but also captures the entire genome, allowing for an exhaustive characterization of genetic AMR markers in a single test. Moreover, such comprehensive genomic information provides a chance to use AI and predictive modeling to emulate culture-based AST and predict which antibiotics represent suitable treatment options. With sequencing costs and turnaround times rapidly decreasing, trying to overcome the shortcomings in the current standard of care through sequencing-based predictive antimicrobial susceptibility testing has therefore firmly emerged as a focus for Aries genetics. Allow me to share with you some of the key challenges, but also the milestones we've met on our mission to revolutionize infectious disease testing next. Once our objective had been set on enabling accurate and fast detection of pathogens and the prediction of AMR, we started to address potential roadblocks. Very quickly, databases for AMR markers emerged as a critical technological component. When we evaluated several public AMR marker databases for their suitability for predictive AST, we recognized a number of shortcomings. Here are two examples. The annotation of AMR markers at the level of antibody classes was too coarse for machine learning, considering that different antibody compounds within the same class are affected differently by any given AMR marker, for example, because of different enzymatic affinities. Therefore, raising the resolution of marker annotation from antibody class to the level of antibody compound substantially improved predictive modeling. In addition, simple rule-based models for individual markers tended to overgeneralize, because multiple genetic factors can, for example, synergistically or antagonistically contribute to the AMR phenotype. We needed to adjust for biology and allow for polyvariate predictive models. Based on these and other findings, we decided to develop ARISDB, a comprehensive AMR knowledge base designed from ground up to enable predictive AST. ARISDB today is a globally leading database of curated AMR markers and information about resistance derived from genotypic and phenotypic data. What sets ARISDB apart from other databases is not only comprehensiveness, but that it also harbors a continuously growing collection of clinical pathogen genomes and associated validated AST measurements that determine how any given pathogen in this database has responded phenotypically in the lab when exposed to a wide range of antibiotics. This additional data layer offers two unique benefits. These genotype-phenotype data associations can be used for machine learning, more specifically to train predictive models that not only discover novel biomarkers for AMR, but that can predict how pathogens respond to therapy based on genomic data, a process we refer to as predictive AST or PASD at Aries Genetics. Currently, the models to predict antibiotic susceptibility are trained as stacked ensemble models on AST results and on a variety of genetic markers, including k-mers, gene and protein annotations, and others, all collected within the ARISDB. This model-based approach can then be complemented by a rule-based approach, taking into account the interplay between multiple resistance mechanisms and antibiotic compounds. As the figure in the middle illustrates, we not only rely on the presence-absence of certain resistance genes, but we also consider changes in non-coding regions of the genome, as well as novel markers that our predictive algorithms have discovered. Allow me to introduce over the next couple of slides how we use ARISDB and predictive modeling to enable a number of sequencing-based solutions for AMR and pathogen detection, and how we validate the performance of these solutions. The Aries solution spectrum today includes a number of next-generation sequencing services and software. Central to our offering is and remains the proprietary ARISDB. This database powers both Aries ISS, our service for bacterial isolate sequencing, as well as the newly introduced Aries UPA. Aries UPA is not in the focus of this presentation today, but I will say that it is a target enrichment NGS panel that comprehensively covers pathogens as well as thousands of genetic markers we have selected from the ARISDB. This assay is currently used in collaboration with 5 early access customers in the public health space in 5 different countries. And finally, the Aries Cloud on the right side is a cloud-based user-friendly interpretation and reporting software, which enables our own NGS service lab and is available to other NGS labs. We demonstrated in several studies the high categorical agreement between standard of care results with the resistance phenotypes we predicted based on NGS data using predictive AST. These studies include, for instance, the first and to our knowledge only multi-center evaluation that is successfully demonstrating the feasibility and performance of predictive AST for 13 pathogens causing lower respiratory tract infections. Some of the study results are shown here, and I apologize for the data-rich nature of this slide. The matrix shows along the horizontal axis the samples for the various pathogens reported by the standard of care. The vertical axis lists the different antibody compounds by compound class that were tested for. The intersections, or the fields in this table, show the categorical agreement in % between Aries predictive AST and culture. If you now exclude all pathogen drug combinations for which pathogens are intrinsically resistant, those are the ones here in blue, and those in gray, for which the standard of care method did not detect any resistance, then the remaining white fields, Then the remaining white fields on the table are the areas in which a direct comparison of Aries PASD and culture-based AST is possible. As the green fields indicate, were we able to demonstrate high concordance, reaching for most of the pathogen drug combinations values significantly above the regulatory threshold for AST devices of greater than 90% and achieving up to 100%. Studies like these are not only important for clinical validation purposes. We also embrace them as they give our data science teams clear indicators on which pathogen drug combinations to focus our efforts on for predictive model development and for database curation. Encouraged by these findings and to help translate this technology into applications, we have more recently validated the entire end-to-end workflow. From sequencing of isolates on Illumina, the de facto standard NGS platform, all the way through the data interpretation via IrisDB and Aries Cloud using our AI-powered bioinformatics pipeline. The workflow performance was very robust in terms of repeatability, reproducibility, sensitivity, and specificity, reaching consistently 100% for pathogen identification, multi-locus sequence typing and genotyping, and values in the high 90s for AMR marker detection. We did actually apply CLIA standards to the validation process, and we published both performance as well as validation protocols in order to make Aries technology portable to other NGS laboratories. So in conclusion, the use of ArisDB, which everyone can access through the Aries Cloud portal, enables the highly accurate and reproducible analysis of bacterial isolates for pathogen identification, typing, and the comprehensive and prognostic characterization of AMR. Both studies that I just mentioned are once more referenced here at the bottom of the slide. The question for Aries Genetics at the moment is, can this technology be applied to further increase the utility and affordability of NGS-based surveillance by relying on faster and lower cost NGS methods? Next, I will give you a glimpse into ongoing work to enable the use of the promising nanopore sequencing technology. As you probably know, antimicrobial stewardship and improved empirically informed treatment with the right antibiotics depends on global surveillance for pathogens in AMR. To make sure the pervasive use of surveillance technologies is also available to low to mid-income geographies, which often act as reservoirs for AMR outbreaks, these technologies must of course be accurate, but they also must be fast, so local clinicians benefit from using them. They must be affordable, they must be deployable and usable in low complexity labs available in these countries. The sequencing platform by Oxford Nanopore Technology, or ONT, satisfies the requirements for speed, low cost, portability, and workflow simplicity. It therefore has the potential to be a platform of choice for fast pathogen identification, AMR detection, and predictive AST. The critical question, however, is if ONT accuracy is sufficient to enable all these applications, or if some, for example, pathogen typing, will be impacted by the accuracy. ONT platforms perform real-time sequencing of long individual DNA molecules. To accomplish this, live enzymes unwind and translocate a single strand through a biological pore and across a membrane. A steady flow of ions through this pore results in a measurable current. And when threading the DNA through this pore, then depending on the base composition that is currently blocking the pore, in other words, the sequence, this flow of ions is more or less blocked, leading to measurable drops in current. These current drops occur in sequence-specific patterns, as some bases lead to stronger or weaker, longer or shorter changes in current. The advantages of this method include low cost, speed, long read length, and a couple of other interesting aspects, such as the potential to directly sequence RNA or to detect chemical DNA modifications such as DNA methylation. A potential issue, however, is that biological pore and DNA translocating enzymes are likely to be not 100% identical across all pores and flow cells. Biological variability is unavoidable and potentially affects base calling quality. We are therefore currently conducting a validation study using the ONT platform, relying on the same 7 ground truth reference strains we have used previously in our published validation studies. This slide outlines the study design. The 7 samples were extracted with suitable DNA extraction methods from 3 different vendors, resulting in a total of 21 DNA extracts. Each extract is aliquoted and sent to 3 partner labs for NGS library preparation and sequencing at an insert size of 10 kb using ONT rapid barcoding kits and the R10.3 flow cells. Raw data are then uploaded to the Aries cloud and subjected to the standard analysis workflow, which consists of QC, de novo assembly, microbial identification, core genome MLST, AMR marker prediction, predictive AST, and plasmid typing. Preliminary data, and these are truly hot off the press, suggest a fair amount of variability in terms of output and read quality between partner labs. Nevertheless, basic nucleotide identity between the resulting genome assemblies and the ground truth remain high, and this is an important prerequisite for downstream analyses. It's also important to note that the number of sequencing errors from ONT, when compared to Illumina, consistently increases 3- to 4-fold. The question we are therefore focusing on is if and to what extent does this drop in base quality affect pathogen identification, the characterization of AMR, and so on. Here you see a deeper dive into sequencing statistics for 2 out of the 7 samples compared between Illumina, here highlighted in blue, and across the partner labs. Again, differences between Illumina and ONT sequencing, as well as variability across partner labs, becomes apparent when looking at sequencing depth, average contact length, number of identified coding sequences, GC content, completeness, and fragmentation. Yet, if you focus on the rightmost chart, then you can see that the quality of the resulting genome assembly remains with tier high very high across the majority of samples and labs, which is a key criterion for good analytical performance on the Aries cloud. So these results were are very encouraging. I will conclude with a preview of the data interpretation on Aries cloud, which, as I mentioned, is very encouraging. Shown here are values for accuracy, measured in % concordance, and reproducibility. In summary, the Aries AI-powered analytics consistently reach 100% for pathogen ID, up to an average of 96% for AMR marker detection, and with up to 100% even higher values for the prediction of phenotypic resistance of the ONT sequenced isolates. First data on plasmid typing suggests that ONT is at least on par with Illumina, which is to be expected from a long read method. However, the verdict is still out on core genome MLST. However, given the fraction of genetic loci that are included in the schemes of core genome MLST that are or appear discordant between the ONT assemblies and known alleles, already hint at MLST as possibly the only Achilles heel we may have discovered for this otherwise very promising platform, allowing for the rapid and cost-effective detection and surveillance of pathogens in AMR. And with that, I'd like to conclude. Thank you for your attention and invite your questions.
And thank you, Dr. Materna, for that informative presentation. We will now start the live Q&A portion of the webinar. And if you have any questions you want to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's get started. It looks like we already have some great questions coming in from our audience members. Dr. Materna, the performance of the isolate sequencing workflow is very impressive. However, I missed your definition of the reproducibility and repeatability. Could you please remind me of how both were defined?
Of course, I'd be happy to. I hope you can hear me well. Still?
Yes.
Okay. Then the repeatability is defined as, well, we're measuring in both cases precision. For the repeatability, we're measuring the precision within runs, whereas for reproducibility, we're measuring the precision between runs.
Thank you so much. And our next audience member says, we're excited to hear that Aries Genetics is publishing validation protocols for the isolate sequencing workflow to support other labs in the adoption of NGS. We are planning to establish NGS capabilities in our lab next year. Do you offer some kind of services that would help labs with the setups of this or similar flows?
That's an interesting question. Thank you for asking that. The exact answer to this question depends a bit on the exact needs, but of course the reason for publishing performance results and validation protocols for the entire workflow, so from sequencing all the way to analysis, is of course to make it easy for any lab to adopt isolate analysis or predictive AST within their own NGS facilities, and to produce the results of the same quality by using the Aries Cloud for data analysis. Now, if your lab, for instance, pursues CLIA registration or CAP accreditation, then the validation burden remains with your lab. But what we offer is, for instance, parallel sequencing of validation samples in our lab and the client lab to help detect and resolve any discrepancies pre-validation.
Thank you so much. Now, AMR profiling and predictive AST, especially in samples we collect for infection prevention, is very interesting, and we would like help. And would it help us to improve empirical selection of antibiotic treatments? How can our microbiology lab access the Aries DB?
The Aries DB is easily accessible through the Aries Cloud. Actually, one of the reasons we've developed the Aries Cloud is to offer easy access to this database. Aries Cloud is a very user-friendly and web-based analysis and reporting portal, which, by the way, is used by our own NGS service lab and is designed such that other labs as well can completely automate the entire analysis and generate reports. For example, for isolate sequencing, the analysis includes, for instance, data QC, de novo genome assembly, pathogen identification, MLST or multi-locus sequence typing, and core genome MLST, and of course the very comprehensive AMR profiling and predictive AST that the Aries DB is known for.
Thank you so much, Dr. Materna. And I also want to thank our audience members for these great questions coming in. Has Aries investigated the PacBio platform for long read?
Very good question. Another very interesting single molecule long read platform. No, not in any extensive validation studies yet. That of course is something that could be of interest. I'd like to point out that PacBio has been around, I believe, somewhat longer than the platform we're investigating now. However, is less portable and is therefore and also more costly, and is therefore less suitable for deployment in a low to mid-income geographies, which as I mentioned are very important for surveillance efforts, as these countries are often most heavily impacted by AMR outbreaks.
Thank you so much. And what is next for Aries, and how would we participate if we are interested in this technology?
Another wonderful question. What's next for Aries is, of course, to offer access to the database through the Aries Cloud, which is a software as a service model. But we have established a Vienna-based NGS service lab in which we support customers in clinical microbiology with surveillance needs, but also customers in the pharmaceutical space who are developing novel antibiotic therapies with contract research or with standard sequencing of isolates or native patient samples. Something that I did not mention today, but I'll use this opportunity, is that we also explore the rich content of genetic markers for AMR in the Aries DB in order to develop a target capture or target enrichment based NGS assay suitable for the culture-free detection of pathogens in AMR in native patient or environmental specimen. So if you're interested or in need of any of these solutions, please don't hesitate to get in touch, and you can reach us, for example, through email to services@ariesgenetics.com.
Thank
that
you,
is spelled Aries dot genetics. Sorry, hold on 1 second. Let me quickly spell out the rather long email address for you. It is services@aries-genetics.com.
Thank you so much, Dr. Materna. We have time for one more question. Have you tried SNP analysis instead of CG-MLST?
Our SNP analysis. Yes, that's a very good question. I believe these two methods can serve slightly different purposes, and we use both. The routine pipeline that we perform on any isolate that we receive includes MLST and core genome MLST. SNP analysis, and this is for those of you who are not that familiar with this type of analysis. This analysis will genotype isolates or strains of a certain pathogen and return a nomenclature that allows epidemiologists or clinical microbiologists to take note of a pathogen that has been detected in the network and associate it with maybe an ongoing outbreak or recognize it as a new outbreak. So this nomenclature is very useful to quickly identify what is going on. SNP analysis is actually more powerful, as you're really using the entire genome and the acquisition of individual point mutations that occur from the initial outbreak of this pathogen and then accumulate throughout the transmission of this outbreak. And analyzing SNPs for outbreaks is something that we routinely do, however, not in order to accomplish typing, as no nomenclature can be associated easily to the SNP phylogeny that is the result. But it is a very, very powerful tool for highly accurate source tracking. So if you want to establish the chains of the transmissions and trace pathogens either back to the source or as I said, follow these transmission chains, then that is the method that we routinely use when we work with customers in the epidemiology space, for instance.
Dr. Materna, I want to thank you for your time today and for your presentation. Would you like to provide any closing remarks to our audience before we go?
I'd like to thank you all for attending and for making the time and taking the time out of your busy schedules. And I'll look forward to receiving any other questions that should arise at a later point in time, appearing in our inbox. Thanks again, everybody.
And thank you again, sir, for your time and for your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via contact information you provided at the time of registration. And I'd once again like to thank Dr. Materna for his time today and for his important presentation. And I'd also like to thank LabRoots and our sponsor OpGen for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now. Until next time, take care, everyone. Bye-bye.